Welcome students. Last episode we discussed the primary and secondary structure of DNA. In the continuation of this DNA structure, in this episode we discuss the various forms of DNA and other structure of DNA. First of all, the differences between the feature of alternative double helical structures. Oligonucleotides of any desired sequence can be synthesized in sufficient quantity and pure enough to be studied by single crystal X-ray diffraction method. However, even with a high pure material, X-ray crystallography is still a challenging technique. It is often likened to gardening by scientists. Some researchers have a green thumb and others do not when it comes to growing crystals successfully. Structures have been determined for a number of synthetic oligonucleotides, usually 4 to 24 self-complementary bases that fold into double helix under various conditions. Studies have revealed the basic structure of three fundamental types of double helix, that is B-DNA, A-DNA and Z-DNA. In the various forms of DNA, first we see the B form of DNA. B form of DNA is the Watson-Crick form of double helix that most people are familiar with. They proposed two strands of DNA, each in a right-handed helix, wound around the same axis. The two strands are held together by hydrogen bond between the bases in anti-conformation. The two strands of the duplex are anti-parallel and coiled. The nucleotides arrayed in a 5' to 3' orientation on one strand align with the complementary nucleotides in the 3' to 5' orientation of the opposite strand. The bases fit in the double helical model if pyrimidine on one strand is always paired with purine on the other strand. From Chargaff rule, the two strands will pair A with T and G with C, that is adenine with thymine and guanine with cytosine. This pairs a keto base with an amino base, a purine with a pyrimidine. Two hydrogen bonds can form between adenine and thymine and three hydrogen bonds form between the guanine and cytosine. B form DNA are the complementary base pairs. The base pairing scheme immediately suggests a way to replicate and copy the genetic information. The width between the base pair is 34 nanometer and per turn 3.4 nanometer and around 10 base pair per turn, and the diameter is 9 nanometer. The B form DNA has a helical pitch of 34 degree to -6 degree base pair tilt and the 36 degree twist angle. The next one is A-DNA. The major differences between A form and B form nucleic acid is the conformation of deoxyribosugar ring. It is in the carbon second atom endo conformation for B form, whereas it is in the carbon 3 atom endo conformation in the A form. A second major difference between the A form and B form nucleic acid is the placement of base pair within the duplex. In B form, the base pair are almost centered over the helical axis, but in A form, they are displaced away from the central axis and closer to the major groove. They result in the ribbon-like helix with a more open cylindrical core in A form. The result is a ribbon-like helix with a more open cylindrical core in A form. The A form DNA is right-handed helix with the 11 base pairs per turn and the 0.26 nanometer axial rise with the helical pitch of 28 degree and the base pair tilt is 20 degree. The rotation is 30 degree angle twist with the 2.3 nanometer helix in diameter. The next one is Z-DNA. Z-DNA is a radically different duplex structure with the two strands coiling in left-handed helix and a pronounced zigzag pattern in the phosphodiester bond. Hence, it is the name Z-DNA. Z-DNA can form when the DNA is an alternating purine pyrimidine sequence such as GC, 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 and indeed the G and C nucleotides are in different conformations leading to the zigzag pattern. The big difference is at the guanine nucleotide. It has the sugar in the third carbon atom of endo conformation like A form of DNA and in the contrast to B form of DNA, and the guanine base is in the syn conformation. This places the guanine back over the sugar ring in contrast to the usual anti-conformation seen in A and B form of nucleic acid. The duplex in zDNA has to accommodate the dissertation of this guanine nucleotide in the syn conformation. The cytosine is the adjacent nucleotide of zDNA is in the normal carbon second atom endo anti-conformation. The zDNA is discovered by Rich, Nordin and Wang in 1984. It has anti-parallel strands as like in the B form of DNA. It is long and thin as compared to the B DNA. It has 12 base pair per turn with the axial rise of 0.45 nanometer at the helical pitch of 45 degree and 7 base per tilt. It has the minus 30 degree twist angle and the helicals is 1.8 nanometer in diameter. Various characteristic features of different forms of DNA. The character orientation is right-handed in both A and B DNA, whereas it is left-handed in zDNA. The major groove of A DNA is deep and narrow, while B DNA is moderate depth and wide, whereas it is very shallow, virtually non-existent, sometimes called a single groove. The minor groove in A form of DNA is shallow and broad, that is superficial, whereas it is moderate depth narrow in B DNA. In zDNA it is very deep and narrow. The bases per turn is 11 for A DNA and 10.5 for B DNA, whereas it is 12 in zDNA. The conditions for the formation of A DNA is low humidity with the high salt concentration, and for the B DNA is high humidity with the low salt concentration. The formation of zDNA is catalyzed by high concentration of magnesium chloride, that is greater than 3 molarity, and sodium chloride or ethanol in the presence of methylated cytosine in high humidity and low salt. Next one is, what are the different conditions favoring A form, B form and Z form of DNA? Whether a DNA sequence will be in the form of A, B or Z DNA, confirmation depend on at least 3 conditions. The first is the ionic or hydration environment, which can facilitate conversion between different helical forms. A DNA is favored by low hydration, whereas Z DNA can be favored by high salt. The second condition is the DNA sequence. A DNA is favored by certain stretches of purine or pyrimidines, whereas zDNA can be mostly readily formed by alternating the purine and pyrimidine steps. The third condition is the presence of protein that can bind to DNA in one helical conformation and forces the DNA to adopt a different conformation, such as protein which bind to B DNA and can drive into either A or Z forms. In living cells, most of the DNA is in a mixture of A and B DNA conformations, with a few small regions capable of forming zDNA. The other rare forms of DNA are cDNA. The cDNA is formed at 66% relative humidity and in presence of lithium ion plus and the magnesium 2 ion plus. It is also right-handed with the axial rise of 3.32 Armstrong per base pair. It has 33 base pairs per turn. The helical pitch is 3.32 Armstrong diagonal to 9.33 Armstrong. Base pair rotation is 38.58 degree and has a diameter of 19 Armstrong, smaller than that of A and B DNA. The tilt of the base is 7.8 degree. The next form is ddNA. The ddNA is a rare variant with 8 base pair per helical turn. These forms of DNA found in some DNA molecules devoid of guanine. The axial rise of 3.03 Armstrong per base pairs and the tilt of 16.7 degree from the axis of the helix. The next one is eDNA, which is referred to as eccentric DNA or extended DNA. eDNA has a long helical axis rise and base perpendicular to the helical axis. Deep major groove and the shallow minor groove is found in the eDNA. eDNA allowed to crystallize for a period time longer. The methylated sequence forms standard aDNA. eDNA is the intermediate in the crystallographic pathway from bDNA to aDNA. Apart from the different forms of DNA, there are some unusual DNA secondary structures are found. Originally, DNA was thought as a static linear string of genetic information. Since the mid-1960s, there has been a rapid expansion in awareness of its heterogeneity and flexibility in form. Through dynamic changes in secondary and tertiary structure, the DNA molecule can regulate expression of its linear sequence information. Unusual secondary structure such as slipped structures, cruciform structures, and triple helical DNA are generally sequence specific. Some of this may be depend on DNA supercoiling. DNA supercoiling provides the necessary driving energy for their formation due to the release of torsional strain. First one is slipped structure. The slipped structures have been postulated to occur at tandem repeats. A tandem repeat in DNA is two or more adjacent approximate copies of a pattern of nucleotides arranged in a head-to-tail fashion. For example, the sequence 5' of TACG, 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 TACG through 3' end contains 4 tandem repeats of TAGC. Slipped structures are found upstream of regulatory sequence, example for gene transcription in vitro. They were characterized by using enzymes that cut phosphodiester bonds in single-stranded DNA but not in double-stranded DNA. It is possible that they have importance of DNA-protein interactions. In addition, number of hereditary neurological diseases caused by the expansion of simple triplet repeat sequences in either coding or non-coding regions. The triplet repeats cause DNA to assume unusual DNA secondary structure, which in turn influence replication and transcription by blocking replication forks and promoting repair. The formation of DNA slipped structures within the long repeating CTG, GGC and GAA strands compared with their complementary strands play an important role in their expansion. The second unusual form is cruciform structure. The formation of short bubble of unpaired single-strand DNA from negative supercoiling can be stabilized by cruciform structure. Cruciform structures are paired stem loop formations. They have been characterized in vitro for many inverted repeats in plasmids, that is small circular DNA, and bacteriophages. Inverted repeat are base sequences of identical composition on the complementary strands. Cruciform DNA can read exactly the same from 5' to 3' on each strand. In other words, the sequence reads the same from left to right as from right to left. Sometimes inverted repeats are referred to as palindromes because of their similarity to a word or phrase that reads identically when spelled backwards, such as rotor or nurse run. Cruciform structures have been visualized by electron microscopy. The DNA become rearranged so each repeat pass with the complementary sequence on its own strand of DNA instead of with the complement on the other strand. Experimental evidence has led to the hypothesis that cruciform structure can act as a regulatory elements in DNA replication and gene expression in various prokaryotic and eukaryotic system. Confirmation of functional role in vivo awaits for further investigation. Triple helix DNA. In a triple helix, a third strand of DNA joins from first two to form triple helix. Triple helix DNA occur at purine pyrimidine structures in DNA and is favored by sequence containing a mirror repeat symmetry. In this type of repeat, the sequence on each side of axis of symmetry are mirror images of each other. The participating third strand may originate from within a single purine pyrimidine tract, that is intramolecular triplex, or from a separate sequence, that is intermolecular molecular triplex. The purine strand of the Watson and Crick duplex associated with the third strand through Gookston hydrogen bonds in the major groove. The original duplex structure is maintained in B-like conformation. Gookston adenine and thymine and guanine and cytosine base pair, named after their discoverer crossed Gookston, have altered patterns of hydrogen bonding compared with the Watson and Crick base pairs. In the Gookston base pair, the adenine base is rotated through 180 degree and the bond to the sugar, and the Gookston guanine cytosine base pair only forms 2 hydrogen bonds compared with 3 in Watson and Crick guanine cytosine pair. Gookston guanine cytosine base pairs are not stable at the neutral pH of cells, that is pH 7 to 8. One of the nitrogens on the cytosine must have a hydrogen added to it for this type of base pair to form, and this protonation requires a lower pH, that is pH 4 to 5. Gookston base pairs have gained importance recently because they are occasionally found in complexes of DNA with anti-cancer drugs, and they shown up in the triple helix associated with genetic diseases. The next one is tertiary structure of DNA. Many naturally occurring DNA molecules are circular with no free 5' or 3' end. Due to the polarity of the strands of DNA double helix, the 5' end of one strand can only join its own 3' end to covalently close a circle. The circular double-stranded DNA is essentially two circles of single-stranded DNA twist around each other. Such circular DNA molecules often become overwound or underwound with respect to the number of complete turns of the DNA double helix. This DNA can then become supercoiled under torsional stress. Supercoils are twisted three-dimensional structure which is more favorable energetically. Supercoiling of DNA can be visualized by electron microscopy and occurs both in vivo and in vitro. Supercoiling of DNA. Considered a double-stranded linear DNA molecule of 10 complete turns with 10.5 base pair per turn. If the end of the DNA molecule are sealed together, the result is an energetically relaxed circle that lies flat. Since each chain is seen to cross the other 10 times, this relaxed circle has a linked number of 10. But if the double helix is undergone by one full form by one full turn to the left and then the ends are sealed together, the result is a strained circle with 11.67 base pair per turn, where linking number is 9 and the twist is 9. Once the two ends become covalently linked, the linked number cannot change. Generally, changes in the average number of base pairs per turn of a double helix will be counteracted by the formation of an appropriate number of supercoils in the opposite direction. In an example, one negative supercoil is introduced spontaneously, re-establishing the total number of original turns of the helix. Over-twisting of the double helix usually leads to positive supercoiling. Another example is if the double helix is overwound by one full form to the right and then ends are sealed together, the result is a strained circle with 9.5 base per turn with the linking number 11 and the twist 11. The introduction of one positive supercoil restores the total number of original turns of the helix, that is, the twist is 10 and the linking number is 11. The supercoil state is inherently less stable than relaxed DNA. The stress present within the supercoiled DNA molecules sometimes leads to localized denaturation in which the complementary strand come apart in a short section. This has important implication for cellular process such as replication and transcription. What are the significance of supercoiling? Virtually all DNA with both prokaryotic and eukaryotic cells exist in the negative supercoil state. If these domains are unrestrained, that is not supercoiled around DNA binding proteins, there is an equilibrium between tension and unwinding of the helix. If the supercoils are restrained with the proteins, they are stabilized by the energy of interaction between the proteins and the DNA. The supercoiling plays an important role in many genetic processes such as replication, transcription, and recombination. Negative supercoiling puts energy into DNA. Underwinding makes it easier to pull the two strands of the double helix helix apart. Therefore, negative supercoiling makes it easier to open replication origin and gene promoters. The potential energy in the supercoils also promote formation of unusual DNA secondary structures like cruciforms. In addition, it is possible that bDNA to zDNA transition is triggered by increased negative supercoiling. This is because switching a portion of DNA from a right-hander to left-hander helix relaxes the strain imposed by the negative supercoils, since the twist in a portion of DNA has been reversed. Positive supercoiling occurs ahead of replication forks and transcription complexes. Positive supercoiling makes it much harder to open the double helix and therefore blocks essential DNA processes. Supercoiled DNA is a well-characterized feature of the circular genome of some small viruses, for example bacteriophage PM2. The genome of this bacteriophage can exist as a relaxed circle or a supercoiled circle which has a twisted appearance. This is the native form in vivo. In bacteriophage, it has been shown that relaxed circular DNA correlates with the reduced activity in replication and transcription, whereas negative supercoiling leads to increased activity in replication and transcription. Similarly, bacteria have large circular genomes that can form independent DNA loop domains. A protein complex holds each domain in place to form subcircles with an average size of nearly 40 kilobase pair. These domains may form supercoiled structure of importance for replication and transcription. Supercoiling also greatly facilitates chromosome condensation in bacteria. The significance of supercoiling in eukaryotic is not as readily apparent as in bacteriophage or bacteria. There are some examples of increased transcription with negative supercoiling, but it is difficult to study conclusively because eukaryotes have such large complex genomes. In addition, replication and transcription process themselves generate DNA supercoiling. Figuring out the mechanism by which DNA supercoiling modulates genetic process poses a challenge. That's all about the various forms of DNA. In comprising this episode, the major different forms of DNA are bDNA, aDNA and zDNA. Apart from that B, A and zDNA, there are also rare forms of DNA which are cDNA, ddNA and eDNA. Some of the unusual secondary structures were found in DNA are slipped structures, cruciform structures and triple helix DNA. The triple helix DNA with the Gookston base pairs play an important role in the anti-cancer drugs and they are shown up in triple helix associated with genetic diseases. The tertiary structure of DNA is supercoiling of a DNA which has a major and important significance in the most of the prokaryotic and eukaryotic cells. In addition, replication and transcription process themselves generate DNA supercoiling, which modulates the genetic process as a challenge. Thus, we concluded this episode and we will discuss a new topic, that is the stability of DNA, in the next episode. Thank you.